Welcome back to Dreamcatcher. Today you're going to hear chapter 10, that is cell cycle and cell division. Growth and reproduction are the characteristics of cells, indeed all the living organisms. All cells reproduce by dividing into dividing with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In another word, such cycle of growth and division allows a single cell to perform a structure consisting of millions of cells. 10.1 Cell cycle. Cell division is a very important process in all living organisms. During the division of a cell replication and cell growth also takes place. All these processes, cell division, cell replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes synthesizes the other constituents of the cell and eventually divide into two daughter cells, and it is termed cell cycle. Although cell growth is a continuous process, DNA synthesis also occur only during one stage in the cell cycle. The replicated chromosomes are then distributed to daughter nuclei of a complex series of events during cell division. These events are themselves under genetical control. So, the phases of cell cycle. A typical eukaryotic cell cycle is illustrated by a human cell in a culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for an example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases, interphase and M phase. The M phase represents the phase when the actual cell division or mitosis occur, and the interphase represents the phase between two successive M phases. It is significant to note that 24-hour average duration of the cell cycle of a human cell division proper lasts for only an hour. An interphase lasts for more than 95% of the duration of the cell cycle. The M phase starts from the nuclear division corresponding to the separation of the daughter chromosomes, that is karyokinesis, and usually ends with the division of cytoplasma known as cytokinesis. The interphase, though called the resting phase is the time during which the cell is preparing for division. By undergoing both cell growth and DNA replication in an orderly manner, the interphase is divided into three phases: G1 phase, S phase, and G2 phase. G1 phase corresponds to the interval between mitosis and initiation of the DNA replication. During G1 phase of the cell, which is metabolically active and continuously grows but does not replicate its DNA. S or synthetic phase marks the period during which cell synthesis or replication takes place. During this time, the amount of DNA per cell doubles, but the initial amount of DNA is denoted by 2n and it increases to denoted as 2c, then it is increased to 4c. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosomes at G1. Even after S phase, the number of chromosomes remain constant, that is 2n. In animal cell, during S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasma. During the G2 phase, proteins are synthesized in preparation for the mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, that is heart cell and nerve cell, and many other cells divide only occasionally as needed to replace cells that have been lost because of the injury or cell death. These cells that do not divide further exit G1 phase to enter an inactive phase called G0 of the cell cycle. Cells in the stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where cell cycle divide by mitosis. For example, the male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. Okay, M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all the components of the cell. Since the number of chromosomes in the parent and progeny cells in the same, it is called equational division. Though for convenient, mitosis had been classified or divided into 4 stages of nuclear division, that is karyokinesis. It is very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase and telophase. In prophase, I'll simplify that. In prophase, if there is initiation of condensation, then the centriole move towards the pole, where the nuclear membrane, nucleolus, all those components disappear and the ester fibers prophase is the initiation of the condensation. The centriole moves to poles, then the nuclear membrane and nucleolus start disappearing. In metaphase, the chromosomes are very thick, and this is the stage which is best to study, where spindle fibers or the formation of the kinetochore, which serves at the site of attachment of spindle fibers. So, and then anaphase, the chromosomes divide, then the centromere move towards pole. It is the shortest stage. Okay. Then telophase is the last stage, which is the reverse of the prophase, where nuclear membrane, nucleolus, all other reappears and the spindle fibers disappears. Okay. Then about the significance of mitosis. Mitosis or equational division The growth of multicellular organism is due to mitosis. It restores the nucleocytoplasmic ratio. Then meiosis is the reduction division. Meiosis is divided into two, meiosis 1 and meiosis 2. Meiosis 1 contains prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 contains prophase 2, metaphase 2, anaphase 2, and telophase 2. Then the meiosis 1 is again classified into 4 types: leptotin, zygotin, pachytin, diplotin, and diakinesis. In leptotin, the chromosomes leptotin, it forms coiled to chromosomes. In zygotin, these homologous chromosomes become so closer and the pairing takes place. Bivalent means the synapsed chromosome, which is 4 number. Pachytin is the crossing over because there is exchange of chromosomes. And recombinase is an enzyme. The crossing over is also called an enzyme mediated process, and the enzyme involved is called recombinase. Diplotin is the crossover chromosomes which repel each other and terminalization of the chiasmata takes place. Diakinesis means the nuclear disappears. The nuclear disappears. And that's what. Meiosis 2 is so similar with mitosis. Prophase 2, where meiosis 2 resembles a normal mitosis. Meiosis 2, anaphase 2. In anaphase 2, the splitting of the centromere takes place and allowing towards opposite poles. Telophase, there are 4 haploid daughter cells. Thus, what are the significance of meiosis? Means it produces haploid gametes, which is half number of chromosomes, maintain constant number, and there is a genetical variation. The variation is due to crossing over. That is the reason for the evolution too. And then few more questions are there, that is NCERT questions. Except that everything is finished. So that's it. Thank you and subscribe to my channel. Thank you.